Another day, another knowledge to quench. Let us normalize learning every single day. I am Gideon, and together with our learning companions, friends, and educators, let's rebuild a nation full of literate students whom develops the future of the world. Good day, class! How are you? I am glad to meet you for today's meeting. I am delighted to have you for today's learning opportunity session. And for today's discussion, we will talk about the steps in DNA replication and protein synthesis in an easy way by utilizing diagrams and charts. But before that, let's first take a look at the objectives for today's learning opportunity session. In the end of this session, the student should first be able to learn the steps of DNA replication and second, the steps in protein synthesis. The third one, students should also be able to diagram the steps of DNA replication and protein synthesis. Hair, fruits, animals, insects, and even human. All living things have DNA. DNA is the molecule inside cells that contains the genetic information responsible for the development and function of an organism. But did you know how the process of DNA replication takes place? I bet not, because we are here to explore it. But before taking on to the exact topic, let us first have a quick review about the lesson DNA replication. The brief history of DNA replication. DNA replication was first found up when the two scientists named James Watson and Francis Crick realized that complementary base pairing provided a way for DNA to copy itself. This base pairing could allow a new strand to be built on an old strand. Realization of these two scientists only proves that DNA was made up of two chains of nucleotide pairs that encode the genetic information for all living things. So what DNA replication is totally about? DNA replication is the process by which a double-strand DNA molecule is copied to produce two identical DNA molecules. Replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as the parent cell. But why do DNA needs to replicate? DNA replication plays an important role in the growth and renewal of cells. Growing organisms are constantly creating new cells as they develop into a larger body. And over time, some cells can become damaged, grow old, or die. So to keep your body functioning properly, it's important that these cells are quickly replaced with new ones. Cells accomplish to this renewal and growth through the process of cell division, in which one cell splits in half to form two new cells. In order for a cell to divide, it must first make a copy of its own DNA, which is the genetic code it needs to function properly. It is very important that your DNA is replicated accurately, with new cells receiving an exact copy of your genetic sequence. So to make it simple, it is essential for DNA to replicate as they tend to save the body from being redundant to being balanced. It is also an essential process in able to cell division to take place. So, I bet you have been recalled on how, when, and what is DNA replication. Because now, get your gear and let us analyze the steps in DNA replication.
Steps in DNA replication consist of four. In the diagram shown, DNA replication undergoes replication formation, primer binding, elongation, and termination. This process is required for the replication of DNA happen. The first step is the replication or fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. DNA has four bases called the adenine, thymine, cytosine, and guanine that form pairs between the two strands. Adenine only pairs with thymine and cytosine, only binds with guanine. In order to unwind DNA, these interactions between base pair must be broken. This is performed by an enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between pairs to separate the strands into a Y shape known as replication fork. This area will be the template for replication to begin. DNA is directional in both strands, signified by a 5 and 3 end. This notation signifies which side group is attached to the DNA backbone. The 5 end has a phosphate group attached, while the 3 end has a hydroxyl group attached. This directionality is important for replication as it only progresses in the 5 to 3 direction. However, the replication fork is bidirectional. One strand is oriented into 3 to 5 direction leading strand, while the other is oriented 5 to 3 leading strand. The two sides are therefore replicated with two different processes to accommodate the directional difference. The second step is what we call the primer binding. In this step, the leading strand is the simplest to replicate. Once the DNA strand have been separated, a short piece of RNA called a primer binds to the 3 end of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzyme DNA primase. A primer must be synthesized by an enzyme called primase, which is a type of RNA polymerase, before DNA replication can occur. The synthesis of a primer is necessary because the enzyme that synthesize DNA, which are called DNA polymerases, can only attach new DNA nucleotides to an existing strand of nucleotides. Next, which is the third step, is the elongation. Enzymes, known as DNA polymerases, are responsible creating the new strand by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria such as E. coli, polymerase 3 is the main replication enzyme, while polymerases 1, 2, 4, and 5 are responsible for error checking and repair. DNA polymerase 3 binds to the strand at the site of the primer and begins adding new base pairs, complementary to the strand during replication. In eukaryotic cells, polymerases alpha, delta, and epsilon are the primer polymerases involved in DNA replication. Because replication proceeds in the 5 to 3 direction on the leading strand, the newly formed strand is continuous. The leading strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerases then adds pieces of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjointed. And lastly, to complete the process, we need the step that is known as the termination. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strand. These primers are then replaced with appropriate bases. Another exonuclease proofreads the newly formed DNA to check, remove, and replace any errors. Another enzyme called DNA ligase joins Okazaki fragments together, forming a single unified strand. The ends of the linear DNA present a problem as DNA polymerase can only add nucleotides in the 5 to 3 direction. The ends of the parent strands consist of repeated DNA sequences called telomeres. Telomeres act as protective caps at the end of the chromosome to prevent nearby chromosomes from fusing. A special type of DNA polymerase enzyme called telomeres catalyzes the synthesis of telomere sequences at the ends of the DNA. Once completed, the parent strand and its complementary DNA strand coils in the familiar double helix shape. In the end, replication produces two DNA molecules, each with one strand from the parent molecule and one new.
Fork formation, primer bonding, elongation, and termination. Repeat. Fork formation, primer bonding, elongation, and termination. Aha! You became familiar with these terminologies. Good thing because it's a sign that you're getting something from what we are tackling. To make it more simple, let's take a look at the summary of the steps in DNA replication. So to make it more simple, just take a look at this. First, hydrogen bonds between base pairs are broken, and then second, DNA unwinds replication bubbles form. Third, free nucleotides pair up with their respective bases. Then after it, DNA polymerase proofreads each new strand of DNA, and lastly, two double helices are now present. As simple as that. So far so good. Good job daw. Kidding aside. Anyways, I hope our learning companion taught you so well because now we will move on to the next topic in line with the first time, which is the protein synthesis. So what is protein synthesis? Protein synthesis is the process in which cells make proteins. It occurs in two stages, transcription and translation. Transcription is the transfer of genetic instruction in DNA to mRNA in the nucleus. It includes three steps: initiation, elongation, and termination. So what are you waiting for? Let's go and explore another adventure.
Just like what Gideon said, protein synthesis have two stages, namely transcription and translation. But first, let us talk about transcription. Transcription is the transfer of genetic instruction in DNA to RNA in the nucleus. It includes three steps: initiation, elongation, and termination. After the RNA is processed, it carries the instructor to a ribosome in the cytoplasm. Translation occurs of the ribosome, which consists of RNA and proteins.
transcription is carried out by an enzyme called RNA polymerase and a number of accessory proteins called transcription factors. Transcription factors can bind to specific DNA sequences called enhancer and promoter sequences in order to resolve RNA polymerase to an appropriate transcription site. DNA transcription is the process by which the genetic information contained within DNA is rewritten into messenger RNA by RNA polymerase. This RNA then exits the nucleus, where it acts as the basis for the translation of DNA. By controlling the production of RNA within the nucleus, the cell regulates the rate of gene expression. Likewise, with the DNA replication, protein synthesis, mainly transcription stage, consists of three different steps, which are initiation, elongation, and termination. Looks so relatively same with DNA replication, right? The first step in transcription is initiation. Initiation transcription is the transfer of genetic instructions in DNA to RNA in the nucleus. After the RNA is processed, it carries the instructions to a ribosome in the cytoplasm. Translation occurs at the ribosome, which consists of RNA and proteins. Next is the elongation. Elongation is the stage when the RNA strand gets longer thanks to the addition of new nucleotides. During elongation, RNA polymerase walks along one strand of DNA known as the template strand in 3 to 5 directions. And lastly, termination. Transcription termination occurs when a transcribing RNA polymerase releases the DNA template and the nascent RNA. Termination is required for preventing the inappropriate transcription of downstream genes and for the recycling of the polymerase. Remember that in DNA replication, there are 4 steps, but in transcription, there are only 3: initiation, elongation, and termination. Keep that in your mind.
Partner Shella tackled about transcription, so I'll be heading on translation, the second stage of protein synthesis. So what is translation? Translation is the process of translating the sequence of messenger RNA molecule to a sequence of amino acid during protein synthesis. The genetic code describes the relationship between the sequence of base pair in a gene and the corresponding amino acid sequence that it encodes. Additionally, translation, the synthesis of protein from RNA, hereditary information is contained in the nucleotide sequence of DNA in a code. The coded information from DNA is copied faithfully during transcription into a form of RNA known as a messenger RNA, which is then translated into a chain of amino acid. Amino acid chains are folded into helices, zigzag, and other shape of form protein, and sometimes associated with other amino acid chain. Moreover, translation takes place in ribosome complex particles in the cell that contain in RNA and protein. In prokaryotes, organisms that lack a nucleus, the ribosome are loaded onto the mRNA while transcription is still ongoing. The mRNA sequence is read 3 bases at time from its 5 and towards its 3 end, and 1 amino acid is added to the growing chain from its respective transfer RNA or the tRNA until the termination codon, normally UAG, UAA, and UGA, where U, A, and G represent the RNA bases uracil, adenine, and guanine, and respectively. Special release factor associated with the ribosome in response to these codons, and the newly synthesized protein, tRNAs, and the mRNA are dissociate. The ribosome then become available interact with another mRNA molecule. It's look like mRNA are taking its spotlight, so we are here to give it to them. Let's tackle about mRNA. mRNA is a single-stranded molecule that carries genetic code from DNA in a cell's nucleus to ribosome and the cell protein-making machinery.
Messenger RNA is a single-stranded RNA molecule that is complementary to one of the DNA strands of gene. The mRNA is an RNA version of the gene that leaves the cell nucleus and moves to the cytoplasm where proteins are made. During photosynthesis, an organelle called a ribosome moves along the mRNA, reads its base sequence, and uses the genetic code to translate each 3-base triplet or codon into its corresponding amino acid. Messenger RNAs, also known as mRNA, are one of the types of RNA that are found in the cell. This particular one, like most RNAs, are made in the nucleus and then exported to the cytoplasm where the translation machinery. The machinery that actually makes proteins bind to these mRNA molecules and reads the code on the mRNA to make a specific protein. So in general, one gene, the DNA for one gene can be transcribed into an mRNA molecule that will end up making one specific protein.
Good job! You're getting into it. If protein synthesis and its steps aren't that clear enough yet, I'll discuss you the summary. Protein synthesis is the process in which cells make protein. It occurs 2 stages, transcription and translation. Transcription is a transfer of genetic instruction in DNA to mRNA in the nucleus. It includes 3 steps: initiation, elongation, and termination. After the mRNA is processed, it carries the instruction to a ribosome in the cytoplasm. Translation occurs at the ribosome, which consists the rRNA and the protein. In translation, the instruction in mRNA are read, and the tRNA brings the correct sequence of amino acid to the ribosome. Then the rRNA helps bind from between to the amino acid, producing a polypeptide chain. A polypeptide chain is a synthesized. It may undergo additional processing to form the finished protein.
Jumbo hotdog, na kaya nyo ba to? Wang nakaya naman! It seems like you're getting really good at listening as times goes by. I can see that you're into like this. I hope you had gained enough knowledge for today's learning opportunity session. We are really pleased to discuss and help you with this topic. Another day, another knowledge that quench. Let's normalize learning something new every single day. Again, I am Gideon Via Costa, and together with our learning companions, friends, and educators, Let's rebuild the nation full of literate and educated students whom develops the future of the world. Thank you so much for listening. Until we meet again. Fighting!
Yeah, 2, 3, 4. Baby, you can get on my boat and make it rock, yes.